Welcome to the GEOTOP webinar. Today we have Dimos Gkountaroulis who will tell us about Knotoids and protein structure. Take it away, Dimos.
Hey, thank you, Duwit. Thanks for inviting me, first of all. I will give you a primer of the theory of Knotoids, and then I will move on to the applications. Also, I will present some results and tabulations that we have with Antistasia, and we have some questions there. So hopefully some of you guys will help us finish this up. And then if we have time, I will show you some of the computational tools that we actually developed for analyzing the topology of Knotoids, in the sense that we have like a set of tools that actually compute Kaufman bracket polynomials and this kind of stuff. But also we can do it in 3D for curves in space, proteins, and all of that. And if we have even more time, I will present some results on actually how we can apply knot theory to the study of chromosomes and how they are entangled and such. Okay. So let's start with Knotoids. Well, the overview is that I will present the basics of Knotoids, then I will show some results on open knotting in 3-space, and then the meat of this talk is part 3, where is the Knotoids and protein structure, how we actually apply all this mathematical toolset to analyze proteins, and in general curves in space, but yeah, I'm working with proteins at least until recently. And then, of course, the software, if we have time. So let's start with the Knotoids. So the idea here is that we want to define a knotted object that is open-ended. Okay, so let's start by drawing a trefoil knot, and somehow let's give up at some point on finishing the job, so we don't finish it and we have something like this. From knot theory point of view, I mean, there is nothing that tells us that this is not trivial. I mean, we can pull the endpoint somehow and we can make it trivial. Okay, so there is no way that we can actually draw something on a paper that is open-ended and it's not trivial in this setting. So, however, if we apply a condition, the condition would be like, don't allow the endpoints to cross over or under any other part of the diagram. Then we are getting somewhere. What we are doing is that we are fixing the endpoints of the diagram in the region that they lie. Okay, so we have one endpoint that it can never escape this region, and we have one other endpoint here that it cannot escape the bigger, the outer region of the diagram. So somehow the topology now is set. So this is knotted. It's a knotted-like object. So what I gave at this point is an intuition of how the definition of a Knotoid diagram would be. And it's more or less the same like a knot diagram. So we have finitely many double points, okay, that they have the information over and underpassing. These double points are different than the endpoints of the diagram. The difference is that we don't immerse S1, but we immerse an open interval, 0, 1. And yeah, we want the image of 0 and image of 1 to not fall on the double endpoints of the double points, because this will not be well defined. And the image of 0 is called the tail of the Knotoid diagram. This is how we decided to call it. And the image of 1 is the head of the Knotoid diagram. And actually, I mean, I didn't say here is that Turaev is the first that introduced all of this machinery here. It's not us, we just apply it to proteins, just to be clear. What else I want to say here is that we want to remember that Knotoids have a natural orientation from tail to head. Okay, so this is also one thing to consider. One step forward is, now that we have a definition, we can actually define equivalence relations. We can do Reidemeister moves, but they have to be away from the endpoints of the diagram. As long as these Reidemeister moves respect the forbidden moves. So we can actually deform the diagram, but we have to keep the endpoints in the region that they lie. Okay, so then we can define isotopy.
Grabación activada.
I'm sorry. Is that a question? I assume it's not. So, for example, now we have equivalence isotopy. I'm sorry, now I lost it a bit. equivalence classes or isotopy classes of Knotoids. And we can clearly see now that these two Knotoid diagrams are not isotopic because there is no possible way of deforming them using the Reidemeister moves and respecting the forbidden moves that we can move from one diagram to the other. Okay. And this is more or less the definition. So a Knotoid is an equivalence class of Knotoid diagrams up to Reidemeister moves and isotopy. And we usually work on a surface. And this surface, most of the time, is either S2 or the plane. And there is an inclusion map, of course, by KR and KS2 are the sets of Knotoids on the plane and on the sphere, on the two sphere. Okay, so there is an inclusion map that we can send the set of all Knotoids of the plane to the sphere, and this is induced by the inclusion, of course, of the plane into S2. I mean, if we take R2 with a point at infinity, we have a S2. We can define it like this, and yeah, any isotopy class of Knotoids in S2 can be represented by a diagram in R2, as long as we push it away from the point at infinity, because then it will not be well defined, and so on and so forth. Also, we have another map that is well defined that we can project from, let's say, from the S2 down to the plane, any Knotoid that we have. And if we take the composition of these two maps, we get the identity map, which means that I, this inclusion map, iota, or I don't know, is a surjection. And we can even observe more that this is not an injection, because there are diagrams that, as we will see in the future, as we will see further down this talk, that they are non-trivial on the plane, but become trivial on the sphere once we lift them on the sphere. Okay, so this is another point that we need to remember. Maybe I will come back if needed here. So now, what's the connection between Knotoids and knots? There is an obvious connection. We can connect the endpoints and we can get a knot. Okay, but this connection has to be in a consistent way. And if we want to work with classical knots, there are two ways to do that. Either we do an underpass closure, so we connect the endpoints with an arc, but this arc has to pass everywhere below. Okay, so we get a knot. Or we do an overpass closure. This is just a matter of choice, okay? Or we do an overpass closure and we get another knot, but we pass every time above. There was a question at some point in a previous talk that I had, is there a way to go over, under, alternate that? But this is not something that will give us a well-defined knot in the end. I mean, it always depends on how you choose to go up and down. So to have a well-defined operation, you just need to connect the endpoints, but you have to be consistent, passing always below or always above. And this gives us a knot on the plane, in S3, wherever you want. Okay, there is another way to go from knots to Knotoids to knots on the torus, in the annulus and then the torus, and I really like this. So let's start with a planar Knotoid. Okay, we can take the double branched cover of that. So we can actually take the endpoints. We have the Knotoid diagram here. We imagine a circle around it. We extend two parallel lines from the endpoint to the disk that surrounds this. We cut, we open, we take two copies of that. We bring in one opposite the other. We do some isotopy, so the ends match, and we just connect, and we get a knot in the annulus. Then we can lift it, and we can have a knot in the torus as well. And this is a tool that we actually use. This is a procedure that we use to do our tabulation, because it's very good. If you actually go to the torus and take a knot invariant there, you can actually distinguish many Knotoids before the double branch cover. Okay. All right. So now about invariance. Since I talked about invariance, there are many invariants that we can define on Knotoids. Many of those are just extensions from knot theory. So we can actually extend the Kaufman bracket polynomial to the case of Knotoids. So we have more or less, everybody knows, I guess, but I will just, for the sake of self-containment, to actually say a few words. So if we have a Knotoid diagram there are two ways that we can smooth the crossings. Okay, there is one way, the A type, where we have labeled opposite regions of a crossing with the same number. So we have 0 here and 1 here. So there is one way that we can smooth it. So we merge the two regions that are labeled with 0, and there's the other way that we can smooth it, where we join the regions labeled with 1. And here are the rules that builds up the Kaufman bracket, actually. So we have a crossing, and we have A smooth one way plus A to the minus 1. These are coefficients, smooth the other way. Then we have the second rule, where we have a loop, a disjoint loop, and a Knotoid diagram, so we can actually pull out the loop and multiply with this factor. And the difference with a well-known Kaufman bracket is that in the end, if we do all this procedure and we smooth every possible crossing, we will end up with a bunch of loops and a single long segment. So we need a value for this long segment. And this long segment, we give it the value 1. And that's the difference with a well-known Kaufman bracket, where the loop gets the value 1. Okay. So let's go ahead and compute something here. So we have these two different situations where they are on the sphere. Okay, I consider them on the sphere. So you can see that on the sphere, I can take this arc. I don't know if you can see it, but I assume that you can imagine. So please let me know if you need more explanation on that. But let's say I'm taking this arc and I'm pushing it around the surface of the sphere past the point of infinity, and I bring it on top. Okay, from here, I bring it here. So these two guys should have the same Kaufman bracket on the sphere. And indeed, if we smooth this one way and then we smooth it the other way, take the corresponding coefficients, we have here a disjoint loop and a segment. We have a disjoint loop and a segment. So we can actually multiply with the factor, and we are done. These two guys have the same Kaufman bracket. But what happens if we are on the plane? On the plane, I don't have the freedom to pull this guy all the way around the sphere and bring it on top. Okay, so these two guys should have different Kaufman bracket on the plane. So this is where we have the loop bracket polynomial. We call it loop bracket. And more or less, So more or less is the same, is exactly the same, but we add one extra rule. If we have a long segment surrounded by k disjoint or concentric circles, let's say, then we give this a value of v to the k, to the number of circles that surround the long segment. Okay. And now if we go and do the same computation again, we see that we have here a disjoint circle that doesn't surround the long segment. Here we do have one that surrounds the long segment. So this will get the value of v, and this will get multiplied the actual factor here. Okay, so we can see that already by this small tweak of the Kaufman bracket, we can actually distinguish Knotoids on the plane and on the sphere. And this is also one example, as we said before, that the inclusion map is not injective, because this is not trivial on the plane, but if you lift it on the sphere, it becomes trivial, because you have this extra freedom to move arcs around the surface of the sphere. All right. So now I will go on and talk about how we did our classification and tabulation efforts to actually tabulate all Knotoids up to 6 crossings on the sphere. This was done also up to 5 crossings by Bartholomew. There is a website, but we wanted to extend it up to 6 crossings because we wanted to do classification of proteins after that, and we knew that the most complex protein had 6 crossings. There was a 6-1 noted protein, so we wanted to do that, and also we said, okay, why don't we do also on the plane? And so let's start by discussing the toolset that we used for that. So we used Gauss codes, more or less how Dowker and Thistlethwaite did it, but we prefer Gauss codes because it behaves better in our setting. So we encode our diagrams. So let's have a a Knotoid diagram here, the Gauss code actually for this diagram goes like this. We choose an orientation. Well, the orientation is set from tail to head, and we assume that the tail is here and the head is here. So we follow the diagram, and we annotate the crossings as we find them. Okay, so 1 and 2. Time that we meet the crossing, we have to say if it is an overcrossing or an undercrossing. And our notation is a plus. We keep 1 as a positive if we go over, and we keep it as a minus if we go under. So we will meet 1 in a positive way because it's overcrossing, minus 2, then minus 1, and 2. Okay, so this is the first part of the Gauss code that gives us the over-under information. But then there is also the information of the sign of the crossing, the parity of the crossing. I assume that maybe I should have given a slide of the definition of the positive and negative slide. But if you imagine that this goes like this, and the undercrossing crosses from right to left, this is a positive crossing. If the underpassing crosses from left to right, this, in our notation, is a negative crossing. So we move around the diagram again, and we see that we cross crossing number 1 from right to left. So we note a plus here, which corresponds to the first crossing. And then we do the same again here, and we see that we go from right to left again, so this is another plus. So we have 1 minus 2 minus 1, 2, so we have the information of the over-under and the parity of the sign of the crossings. Okay, so this is for the sphere. For the plane, we need to add a bit more information. So apart from the crossings, we annotate also the arcs of the diagram. So we have the arc 0, 1, 2, 3, and 4, and we have to annotate which arcs touch the outer region of the diagram, and that's it. So we have a third part in this Gauss code that says 0, 2, and 3. So we can actually know which part of the diagram, because there are many ways that you can deform a diagram in S2 and have different regions outside. Okay. But now you can actually reconstruct it even in R2. You know exactly what you're talking about. You have the full information. Also, I would like to talk about some involutions here that we can define on Knotoids. The first involution, we read it like this. This part of the slide, we read it like this. So if we have the orientation of the Knotoid K, and we have the Gauss code, we can define the reverse of that. So the reverse is just we invert head and tails. So we go the opposite way. And again, in this case, the Gauss code remains the same. Sometimes the Gauss code in the reverse changes, so it's not very stable. But there are 3 other involutions that I would like to discuss here. The first one is the mirror. And this is the mirror image, okay? So if we have the Gauss code here, the mirror image inverts over-under information and changes the parity. The symmetric is something like taking the antipodal, more or less. If you have it on the top of the sphere and then you take the antipodal symmetric on your sphere, which actually maintains the over-under information, but changes the parity. And the rotation is actually changing the over-under information, but keeps the same parity. So these are the 3 involutions that play important role, because if you choose a strong enough Knotoid invariant, like the double branch cover, all of these guys get different values. So you can actually distinguish all 4 of them with a double branch cover. The reverse doesn't change much. Even if the Gauss code changes, these 2 guys will always land in the same isotopy class. They will get the same value of the Knotoid invariant. So we define the involutions. We can actually encode, and I will just go very quickly through that. we can encode the Reidemeister moves with Gauss codes. So we can, if A is a crossing and W is a part of a subword, if we have A with some kind of sign and then we meet it immediately after with inverted signs, then this is a Reidemeister move and I can get rid of it. Same goes for Reidemeister 2 if I have a crossing and then I meet another crossing and they both have the same sign and then I have a subword of the Gauss code, and then I meet them a bit afterwards, but with inverted signs in the same direction. This means I have a Reidemeister move, and I can get rid of them, of these crossings. And also, depends also the orientation of the arc. So even if I meet, again, those two crossings, but with inverted order, again, it's okay. I mean, it's a Reidemeister move. I can get rid of them. And then we have 4 cases of the Reidemeister 3. But I think it's not worth to go in detail about that. But you can. I have everything in detail in our paper on arXiv about this. So I will give the details of the preprint a bit later on. So okay, so we know about the involutions, we know how to encode diagrams, we know how to perform Reidemeister moves, so now we need an algorithm. So we need somehow to sort them and to have an order of that and perform our algorithm. So we will choose the maximum number of crossings that we want to go up, I think in English. Anyway, I will go with it. So let's say that we want to do all Knotoids on the sphere up to 6 crossings. So we will generate all possible Gauss codes on the sphere, all possible combinations of 1, 2, and 3 with minuses for over-under information, and all possible parity combinations. Okay, so this means that I will have a lot of redundant diagrams there because they are not in minimal crossing representation form. But we need that. Okay, we need that to generate our isotopic classes. Then, once I have generated this huge list of all possible combinations, I will impose an ordering, and I will sort this big list by the length of the Gauss code. So Gauss codes that are shorter, they will be higher in our ordering, and the longer Gauss codes with 6 crossings will be on the bottom. Okay, and then we give another ordering of first crossing goes, crossing number 1 comes before number 2 and so on and so forth, but also we give priority to the undercrossing than the overcrossing of each crossing. So the version of a Gauss code that starts with an undercrossing will be higher with the same Gauss code if it had the over-undercrossing information switched. Okay, and then I give also priority to minus parity than the plus. So this is the order that we decided. It's not universal. It's just the convention that we used. I mean, other people can decide some different sorting as long as it is consistent throughout the tabulation. And then, if we work on the plane, if we work on the sphere we done here by step 3, if we work on the plane, we will sort by the length also of the outer region, and then we order also the arcs that touch the outer region of the diagram. So this is nice. We have a nice sorting of all our Gauss codes, and now we start evaluating all these Gauss codes with a choice of knot invariants. We used 2. For the plane, we used the double branch cover, and we evaluated the knots on the torus using the Kaufman bracket on the torus. And we used also something that we called the loop arrow polynomial. Everything is in our preprint, all the details. And for the sphere, we just used the arrow polynomial, which is another invariant that I didn't talk about it, but it's a bit more complicated and didn't want to spend time on explaining the combinatorics of that. Anyway, so we get a partition for each equivalence class, and then within this equivalence class, we try to apply Reidemeister moves to connect all the parts in this equivalence class. If we connect all of them with Reidemeister moves, means that all of them are isotopic, and this is actually an equivalence class, not an equivalence class in quotes. And then we need to find all the involutions, and since we use strong invariants, all of them have different values of the invariant, and we will merge them because the classification was up to involution, so all the involutions for us were the same Knotoid. And we do a final pass of Reidemeister moves in case of we missed anything. So now, if this went over your heads, I will just give a graphic representation. So I have ordered all of my Gauss codes in each isotopy class. So they have the order that we wanted to. And now we start applying random Reidemeister moves between in each isotopy class. So let's say that if I started applying Reidemeister move, I connected all of them in that way, okay, somehow. So everything is a connected graph here. There would be more connections happening, but okay, I choose just one. And the same happens for each one of the three other involutions. So means that if there was an isolated point here, this would mean that there is no way that I could go from here to here with Reidemeister moves, and possibly there could be a case of non-isotopic Knotoids having the same invariance, so we couldn't distinguish it with the invariance that we actually use. But for the sake of this example, let's say that this doesn't happen, so we have one full connected component here, one full connected component here, so on and so forth. And since this classification is up to involutions, then we connect the corresponding involutions from this guy to this guy, from this guy to this guy, from this guy to this guy, and we perform a final pass of Reidemeister moves to see that actually all of this is a connected graph, and these are the biconnected components of the graph. And once we finish, and we have for all equivalence classes, let's say, not isotopy classes yet, we have this situation, we say that our tabulation is complete, and that's it. We show the results. And actually, what we did, we found that in S2, this terminates, and we actually have this result. So we don't have any Knotoids, prime Knotoids, like in prime nodes that have one crossing, because we cannot. There is only one with 2 crossings, 2 with 3 crossings, 4 with 8 crossings. I'm sorry. 1 with 2 crossings, 2 with 3 crossings, 8 with 4 crossings. You see that it's a lot more rich. We have more Knotoids than knots here. We have 24 prime Knotoids with 5 crossings and 161 with 6 crossings. All of them, I mean, if you take all the 121 and close them, somehow you will get to a 6-crossing knot, more or less, most of the times. Some will get to smaller knots, but most of them get in the 6-crossing region. In any case, so you have 156 in total Knotoids on the sphere with up to 6 crossings. On the plane, things get wild. On the plane, we have one Knotoid with 1 crossing. I showed before in the example of plane sphere, we have 6 with 2 crossings, 26 with 3, 154 with 4, and 950 with 5 crossings. So we have a total, and we are crossings 5 crossings here, 1,137. We didn't do 6 crossings because actually we didn't have the computational power at that moment to do 6 crossings Knotoids. So yeah, we stopped here. And also we found the other problem that we have 6 pairs of Knotoids that we actually, on the plane, that we couldn't distinguish, no matter what invariant we used, no matter how long we let our algorithm run. So yeah, these 6 pairs, so 5, 7, 5, 4, 2, 1, and so on and so forth. You can find the details here, and I can actually well, if I click it now, I think I will get out of the presentation mode, but I can actually link it up later if anyone is interested. All of those knotoids, the thing that all of these pairs share is that these two map to the same knotoid on the sphere, but they have a different region of the diagram marked as the outer region. That's the only difference on the plane. On the sphere, they are the same. And we cannot actually find a way to say that, there is, for what we checked, there is no way to go from one to the other using Reidemeister moves. Maybe we missed something. Our software backed out, but yeah, this is a possibility. But I would say that these are different knotoids, and I didn't have the time, honestly, and I didn't, to actually go and to do a rigorous mathematical approach to prove that they are actually different. But I suspect that they are. And I would be happy if any one of you has an idea on how to actually show this, and we can actually work together if you're willing and we have the time to actually finish this up and submit it. This is the conclusion of the classification that we actually use later on the protein structure. So let's talk about. I will take a break here and ask if there are any questions in this part, because now I will not talk about this stuff in the following slides. So just wait 2 seconds.
So Dimos, for those 5 crossing ones that you cannot prove for this are different, they have all the same invariants, and there's nothing you can do any further unless you get a new invariant. Is that what you're saying?
Or someone, yeah. We need a new invariant. I've tried HomeFly. I tried all the versions of the Kaufman bracket, Alexander. On the double branch cover, we used the HomeFly on the sphere, the Kaufman bracket on the sphere. We did several other. Widening numbers is the same. Even rudimentary invariants we used. Case. We missed something, but so far we didn't come up with an intelligent answer to say that, yeah, these are definitely different. Yeah, we will be very happy if someone has a very nice idea, and I'm sure if someone spends some time, I guess they will they will find it. I can link up my email if you want to actually have a discussion later on whoever is interested.
Dimos, I have a curiosity. It's a rather simple question. Why go for knotoids instead of open braids? And there is any reason for not considering braids and open braids in particular? They would equally be useful for this.
Yeah, equally useful for classification of curves in space, open curves of space, or for what?
Also, well, there are techniques applied to open braids, and I wonder if these can be of any help in what you're doing.
The classification of knotoids, or in general?
Well, in general, I wonder if it is any reason to prefer knotoids rather than open braids.
Yeah. The motivation was, first of all, I liked it. But secondly, the motivation was that in protein studies, they get these open-ended knotted structures, and the go-to method is to find an unbiased way to close the curve. And this is there is no rigorous mathematical way of what's the best way to close an open curve. It's an open problem. And I said, what if we don't close the curve? What if there is a way? And there was Knotoids in the way. I mean, you can actually not close the curve, and you can analyze the protein as a Knotoid in the end, which actually turns out to give more information also on the topology of a protein. And from a computational point of view, it's not actually a lot slower than doing NUTs, which is a very important part of the pipeline if you are dealing with big data sets of thousands of proteins. I mean, yeah. I don't know of any tool also that actually does braiding to compare times. I mean, they could be stronger, and there might be a program that actually computes all of this information faster than our pipeline does, and that would be amazing. But I'm not aware of that. It doesn't mean that it doesn't exist, but yeah. But my main motivation was to avoid closing the curve. I wanted to keep it open and analyze it as is. Because biologists, when you tell them, okay, let's close the curve, and they tell you, well, the geometry is different, the curve is open, why do you close it? They don't have most of them don't have the mathematical intuition that to have a knot, it has to be a closed curve to actually analyze it as a knot. And Knotoids do that. I mean, that was the motivation behind that. And there wasn't any tabulation, so I did the tabulation. There weren't any computational tools, so we built the computational tools. I mean, we built the whole pipeline actually from scratch, based on two papers. There was a Kaufman paper, Luke Kaufman, and the first paper by Turaev. And everything else followed.
Mm-hmm. Okay. No, because in magnetic field, in the study of solar physics, people, Mitch Berger, Hornig, and others, use open braids. And this is because just the ends of your Knotoids are almost like fixed on a, if you like, on a surface or a plane. And so you can find techniques to analyze that. And that's why I asked.
I would see them more like a 1-1 tangle than an open braid, honestly. But
sure.
Yeah. I mean, there could be a way that to analyze all of these things with open braids. Actually, Sophia and Neslihan, her PhD student, did a study of braidoids. I mean, how they have some kind of braid structure that if you close it, you get a Knotoid in the end. And they applied it to protein structure, but I mean, there is a theoretical model and no one built a tool for that. So I don't know how fast it is also to compute this kind of stuff.
Because of course you can close a braid with a trivial braid, yeah. And you are not going to modify your knot type. Nevertheless, you adopt a different kind of coding from braid theory to analyze the complexity. That's all.
So yes, this is very reasonable. It's just that I didn't think of it also, honestly.
But it seems to me that, given these unresolved pairs with just a single arc, if you have a braidoid with 2, 3, 4, 5 arcs, that problem's going to get much worse.
I get much worse.
Okay.
Why I want to keep it with a new Knotoid invariant than developing new geometric topology techniques, I guess, to figure out this kind of stuff. But thanks, Renzo. It's reasonable what you suggest. I didn't think of it, honestly.
Okay, thanks.
Hi, Dimos.
Yeah.
Hi. Now that you mention it, is there a relation between Knotoids and tangles?
It's the moment on the sphere. On the sphere, you can actually, when you're. Do I have a Knotoid on the sphere? Let me. Okay. So this is a 1-1. All of them are a 1-1 tangle. You can actually make them as 1-1 tangles, especially on the sphere. On the plane, I mean, on the sphere you can always deform them so one endpoint is always on the outer part. So you can work a bit better with 1-1 tangles. I didn't go that much into this theory, but we were we spent like a couple of days with Luke Kaufman when he was visiting Switzerland to think about this, and we worked only with spherical because of the ease of getting at least one endpoint out. On the plane, you have more weird stuff happening where both endpoints can be very much inside, hidden in the whole complexity of the whole diagram. That I don't know what's going on there. You could define somehow. I think maybe there could be a way to deform it and actually define a tangle there, but I didn't think of that. I didn't go there. My main focus here was to actually find a combination of invariants that would, if I apply them, will just sort them out. I mean, because I use a combination of two invariants. I use the loop arrow polynomial and the double branch cover. So the combination of the two gives me this fine clustering, let's say, into equivalence classes. If I could find a third one that, if I plug this in, splits a bit more this situation, that would be ideal. But I don't know. I'm not sure. I'm not sure how to go from
Thank
here.
you very much.
I mean,
Yeah,
I know a colleague of.
There
is a colleague that thought of
no
it.
close relation. Oh, okay.
Sorry.
I was wondering if the relation between the knotoids and the tangles, or if there is a natural way to construct a tangle from a knotoid in such a way that any knotoid corresponds to just one tangle, and every tangle corresponds to just one knotoid. But probably that's not true, I imagine. But I don't know how close the relation is.
It seems to me like a research proposal. This. I mean, I'm not. I'm not sure if someone thought about that. Honestly, I've been following the literature on knotoids quite close. Maybe I missed a publication, but I don't think that anyone thought on or published something or put a preprint anytime soon on what you're suggesting. So this sounds like an open problem to me. I mean, yeah, could be.
Thank you very much.
So it's the third time that I'm on this slide, so I will just try to go past it and move on to the third part of this talk. So I will talk about open knotting in 3-space. So let's go back to discussing open knots, and we have 3 examples here of a trefoil in space. There is a cut. Intuitively, we know that, I mean, from intuition, from everyday life, we say, okay, this could be knotted, but from a mathematical point of view, they are not. Because there is always at least one way to undo the endpoints. And moreover, if we look something from a different point of view, like if we twist the last one, we see that it's not clearly knotted, it's just like an illusion. So as I said, there are what people do to actually capture this knotting, because we want our results to match our intuition, because we see it's knotted. We just cannot capture it with the available mathematical tools. We need an unbiased way to close the curve. And I have here some examples, and I don't have Christian's method, and sorry about that. But Christian has a very robust method also of closing the curve. One way is like we have the direct closure, which is more or less Christian's method is a blend, is the best of two worlds, like blending these two things that I will talk about. So one way, the intuition here says that we just connect the endpoints, but this sometimes it misses the point if we connect the endpoints, because let's say if we try to connect the endpoints here, we undo the knot. So this is not what we want. If we connect the endpoints here, this is something not this is something trivial, but if we connect the endpoints, we create a node. So this is a hit and miss, more or less. But sometimes, I mean, I don't know how to say this now, but the error in the end, if you do it many times, is not that big. So depends also. There is no wrong way in closing the curve in the end. It depends what you want to do and how fast you want to do it, and what result. If you have a big population of open curves and you want to see what's the percent noted, back of the envelope computation, you will get something with an error here, but the error won't be that big of a deal, I think. Correct me if I'm wrong, Christian. And then we have the center of mass closure here, where there is another way that we have centered the curve in the center of mass, and then we extend rays from the center to the endpoints, and then we connect them when we are far away and we close the curve. And that's a single closure method. Okay, this is a deterministic approach. What I think is most used in the literature is probabilistic methods. So we consider all possible closures of a curve given a method, and in this method I present the infinity closure, and I will explain what all of these colors mean. And then we have a space of closures, and we sample the space to actually see what's going on, and we have a distribution of knot types, and that's the topology of the curve. It's actually the distribution of the knot type, and the predominant knot type is actually the knot type that represents the distribution and characterizes the curve. So if you have, let's say, an open curve that looks like a trefoil, and in the distribution of your sampling of knot types, you get 85% a trefoil, then it's a trefoil most of the time, so you say it's a trefoil. And the infinity closure that I'm presenting here is widely used in protein topology applications. Of course, other methods are used as well. Anyway, used quite a lot, and I like this, and I have implementations for this, so that's why I like it. Probably I'm a bit biased because I have already the tools to actually do this kind of computations. So the infinity closure is the following. So you consider that your curve is centered in a large enough sphere, and this, we can say, it's the convex hull also of the curve, okay, in mathematical terms. Now, we have the endpoints, and each point of the sphere that encloses the curve represents a direction towards which I extend rays from the endpoints, and these rays should be parallel. And once those rays pierce the convex hull or the surface of the sphere, I just connect them. And that's it. And I do this for all possible directions. And once I do this for all possible directions, I evaluate all of those with with a not invariant, and I get what I said, the distribution is the predominant. Yeah, okay, and as I said, the biologists that I've worked with, and they said that, okay, this is good, but we're changing the geometry. How we can actually find a way to not close the curve and measure the topology of that. And actually, we can use knotoids, okay. So why not we project? We take a projection. This is a projection. We have a knotoid. Okay. What happens with the problem of the endpoints? Because now, if we do any ambient isotopy, if we deform the curve in space, if we do anything here, the endpoints will be displaced. We will lose what we measured here. And the answer comes from Luke Kaufman, and he said, okay, why not we put this kind of infinite lines that go through the endpoints of the curve to the plane of projection, and they are there. And every deformation that happens with respect to these two infinite lines. And this is the concept of line isotopy. So if you keep those rails, let's say, imagine this as rails where the beads go up and down, and everything here deforms but never crosses the rail, then you actually capture the topology here. And yeah, that's a way to go. We can actually use that and do topology of open curves and measure proteins. But But if we see it from different points of view, if we do different projections, we get different Knotoid types. Okay, so again, this is a probabilistic approach. So we need to sample all possible projections and get the predominant Knotoid type. And one way to visualize this kind of stuff is the following. As I said before, this is more or less a probabilistic approach. So if we have a curve here, we follow more or less what is done in the infinity closure. We imagine that this is enclosed in a big sphere. Each point of the sphere gives me not a direction toward which I close the curve, but a direction towards which I project and I insert these two rails. And if I do that, okay, I can color code each point of the sphere with the Knotoid type that it gives me. So this is how it looks like. I mean, regions with the same color give me the same Knotoid type. I mean, it doesn't matter which Knotoid type it is here, but we see that we have a big distribution of Knotoid types. The biggest region of the region with the biggest area is the predominant knot type. Okay. So we can actually say that, okay, this, I don't know what kind of color it is. Let's say it's red, but it's not red. Is a 3-1, so this is most of the times a 3-1 because most of the projections give me that. There could be border cases where you have like 45% and 45% of something, so you use both. You say it's half of the times a 3-1, half of the times a 5-2, or whatever. But this is more or less the case. And now, that's the gist of it, okay? This is how we use Knotoids to find the topology of open curves. And of course, now proteins, this is how we apply all of this to proteins. So first of all, proteins are open-ended structures. They are made of amino acids. They are stitched one next to the other. Imagine like a necklace with beads, more or less, in a coarse way. And it has been found so far that 6% of proteins are known to be knotted. They have some kind of knot in their conformation. And that this knotting pattern is preserved across species that is separated by millions of years of evolution. So humans and yeast share a knot in a protein in the same place, more or less. One example. And why is that there? I mean, was it selected? Is it by evolution? Is it an accidental thing that didn't hurt the cell or the protein and kept on through the evolution? We don't know. But there could be some advantages to those proteins that have some knot. And there have been some studies supporting that. There have been some studies against that, but I will talk about the studies that support this kind of hypothesis, let's say. So there are some advantages. For example, if we think about this protein, this human ubiquitin C-terminal hydrolase, has this very weird name, the function of this protein is the following. So when a protein is in the cell and completes its role, it gets recycled, it gets degraded, and it gets recycled within an organ that's called proteasome. To be recycled, it's marked by a genetic marker, okay? Genetic, that's the correct word. Marker, when it's detected by this organ, it sucks the protein in, but the marker doesn't need to go in. So there is this protein that operates at the border of this organ that removes the marker. But since it operates so close to that, sometimes it gets sucked in and it blocks and it creates a mechanical stress there. The knot is tightened, so everything blocks even more, even worse. So at some point, the organ expels the protein back, and this is protected from degradation and continues working, removing markers as is intended to do. So actually, there are some structural advantages here. I mean, the knot tightens, protects from being sucked into somewhere that it shouldn't be. Okay. So let's see this protein. The way that we analyze the proteins is that we trace from each amino acid that makes a protein, we trace a carbon atom there, and we actually make a backbone. We find the backbone of the protein by tracing the carbon atoms there. And we can actually smooth even more, remove, with Reidemeister moves, let's say. There are other more sophisticated moves that, but think of some kind of Reidemeister moves and smoothings that we can do on the curve, and we can get a final smooth structure that actually preserves the topology. And we do what we discussed before. We actually compute the distribution of Knotoid types, and we see what we get out of it. So if we manage to, the classification of proteins using Knotoids may actually give us an insight of what is the role of knots in proteins. So that's why are we interested in that. We want to classify and see what's going on. I mean, if particular family of proteins that does a particular kind of thing is knotted in a particular way, probably this knot keeps something there. It serves some purpose, probably, maybe not. But yeah, so that's the idea. And let's now compare a protein analyzed as a knot, how this is a stereographic projection of the sphere, okay, that has all the closures. So here the protein is analyzed as a knot. We have a big blue area where most of these points, when we took the direction to close the curve, gave us always the same number of the same type of knot. We have smaller regions that gave us a different kind of knot. And now we have the same protein analyzed as a spherical knotoid. So there is a lot of information in the middle that we lost. Okay, so there is something here that didn't exist before. So we lost all of that information. And that's the knotoid on the sphere. If we go to the plane, we have even more information coming up. It's like going from knots to spherical knotoids to planar knotoids. Like we crank up a bit the resolution of our focus, so we see a little bit better. The point is that here there is no wrong way to approach this. It depends always what we want to see. So there is nothing wrong by working with knots if you just want to see quickly what's going on there. But if you want to see the real topology of a protein, I think planar knotoids is the way to go, because you get the full spectrum of all possible entanglements that you can get there. You may get more geometric features and this kind of. And all of this was summarized in two papers, one 2017 and the other one was in 2017, one in Scientific Reports, one in Polymers. One is with Andrei Stasiak and his group, where I was a part of, and the other one was with Luke Kaufman, Sofia Labropoulou, and some members of Andrei's lab. And yeah, so that's the conclusion of how we analyze the topology of proteins using knotoids. And the conclusion is also that we see a bit better. It's like we crank up the resolution, more things there. Now, let's take it a bit further. As I said before, we take a number of projections. We take a sample. We sample the space of projections. So we take a number of them. What's the optimal number of projections? Is it 50, 100, 5,000, 100 million to get the best resolution possible, to get the best outcome? So we worked on that with my colleague Agnese Barbensi. She's a postdoc now at Oxford. She's a Hooke Fellow. And we thought, okay, so let's think about this stereographic projection of the map, okay? So if we have knotoids in neighboring regions, this means that these regions actually differ by a very small perturbation of the projection angle. I mean, you look at one way and then you change it a bit slightly and you get a different knotoid type. So this means that the endpoints somehow get displaced and we do a forbidden move here. So can we define a measure that captures this dynamic forbidden move, this forbidden movement, if you think? So we define the forbidden distance. So given two knotoids, we define the forbidden distance of two knotoids as the minimal number of forbidden moves that we can do across all representations of these two knotoids that we can to transform one knotoid to the other. And then we have a theorem that says, before going to the theorem, then we can actually make tables of this distance, like the Witt and Isabel Darcy did for strand passages. So this is what we did here in this paper. And yeah, we had the set of Gauss codes sitting. So we said, okay, let's do an experimental approach and compute all the F distances between the Gauss codes so we can have some upper bounds. And then let's make up a theorem. And we can abstract some lower bounds for that and see what's going on. So the theorem says that if we have two equivalence classes of Knotoids, and they are up to rotation and inversion, then the following three conditions are equivalent. So if two Knotoids differ by forbidden moves, then their corresponding theta curves, and I will explain a bit here, differ by strand passage by one of the three edges of the theta curves. Now, it's important to say here that we work with Knotoids on the sphere. So what I'm hiding here is that the theta curve is actually the double branch cover of a Knotoid on the sphere. So when you do double branch covering of a Knotoid on the sphere, you get the theta curve. So instead of characterizing Knotoids that differ by a forbidden move, we can very well work with theta curves. Or we can take the constituent nodes of the theta curves, which a constituent node of the theta curve is that you consider only two out of the three curves and you see what node is there. If they are strongly invertible, which is a definition that I will point to you to our paper to actually go and check everything out, then these nodes differ by equivariant Barth surgery. So in the end, what we do is we check if the theta curves differ by strand passage of two of their edges, or if there is an equivariant, non-coherent band surgery that is happening between the corresponding nodes of the theta curves. And these actually help us to build a table of distance of F distance of Knotoids. So we have this. The different coloring means that one was done with theta curves, the other was done, I think the blue is the theta curve, the orange is, this was abstracted by the theta curve, the orange was abstracted by the band surgery, and the rest we couldn't actually say something about this. I don't remember about the red, and the black ones were simulations and theorem agreed, so we didn't have to do anything. And so now, having the F distance, if we take the set of all proteins, analyze them with a number of projections like 50, 100, 500, as we did here in this experiment, 1,000, 5,000, 10,000, we check the average error. And the average error is defined as the error between Knotoids sharing the same border. And we see that with 50 projections, we have almost 15% error. We have 100% error with 12% error with 100 projections. And as we move down, we see that in this region, we are in a good place. And I mean, we have less than 5% error. And we can actually work with either one of those. I mean, we can actually go even better, do more projections, but now we are entering in the computational time problem. So I think a good balance of good approximation and good computational time, if you want to do it in a single machine, okay, because there are servers that do it automatically, but if you want to do a quick pipeline on your machine, I think 5,000 projections is enough to actually sample well your space of distributions. And this is one of the results that we have with Agnes. And this appeared actually this year in the Proceedings of the Royal Society. This is the details of the publication. And one final thing, and I see I have a spelling error here, is our latest result that actually appeared on arXiv yesterday, and I want to quickly talk about it. What time? Okay, so we're a bit over time. Tell me if I need to stop. But so far, we've been using only the predominant knot type of a protein to characterize them. And the question is, can we use the entire distribution and somehow probe indirectly the geometry? And is there a way to compute a distance between those distributions? I mean, if we have a Knotoid distribution for one protein, Knotoid distribution for another, is there a way to say they are that far apart somehow? And the answer comes from the Earth mover's distance, or the first Wasserstein's distance, over a region D. And this is a statistical measure. And also, it's an optimization problem. But in any case, it solves our problem. So the Earth mover's distance can be imagined as the minimum cost of transforming one distribution to another over the same region D. And I have two examples here from random Gaussian data. Okay, and you can imagine each bar here being a pile of Earth. Okay, that's the minimal work that I need to spend to transform and move dirt around to transform one histogram to the other one. And that's the Earth mover's distance. And it's a well-defined metric. It's a metric, actually. And we say, okay, why don't we define Knotoid, which actually our distributions and our region is all possible Knotoids. And the cost function of that is the distance. So what do we get out of that? So we took that and we threw into this all trefoil knotted proteins. And we actually see that, first of all this measure recovers clustering by sequence similarity, meaning that proteins that have at least 30% sequence similarity, which is in biology enough for two proteins to do the same function, they actually cluster, they fall in the same cluster. They cluster very close together. So we have some representatives of each family, and we see that they are clustering very well together. So this is one nice result. So we can use this. Here, you may ask, what is UMAP1 and UMAP2? So UMAP is a unsupervised clustering method. It's more or less a bit of machine learning that actually takes high-dimensional data, builds a manifold, builds some fuzzy topology in a manifold based on that data, and then does a dimensionality reduction, a projection, but with the aim of preserving as much as possible the distance that initial manifold had. The metric of that is preserved in the projection. So we can actually see on a plane with a good accuracy what's going on in the higher dimensional space. Because here, what we did in this part is we took all the pairwise distances between trefoil knotted proteins. So this is what we computed, and it's a big distance matrix. It's 500 something by 500 something. So this is a big space. So we needed to project down to see what we did. And the pairwise distances, they actually cluster together in a nice way. So proteins, as I said before, have at least 30% sequence similarity. They fall together, and that's a nice result. And one final thing is that, can we say anything about folding, since we went that way? So folding, and in this case, there are four theories that a protein could fold. And in all of those theories, one way or another, there is a single loop that it's formed, and there is some kind of threading that's happening afterwards. So then this is how you make your knot, okay? But recently, like I think, now it's been 2 years, I think, but let's say recently, there was a new theoretical model by Erika Flappan that suggests that there is a double loop mechanism that's happening here. And she suggests more or less this kind of pathway. So you have first forming two loops, then you have the blue part of the loop. So you have the movement of the blue part of the loop. So it either pierces the red loop or the green loop flips over, and then you have the final threading and the placement of the loops. And then this is more or less one of the ways that you can actually use this double loop model. And we said, okay, or maybe we can get these geometric features and see what we can do with this Knotoid measure. And what Flapan suggests is that the theoretical model has all the cases that you come up with are these. And you have, it's a bit complicated, but the idea is that L and R is which part of the green loop and the red loop comes first over the blue one. So the left and the left, you see here it's LL. So, and you read it from inside out. So it's red and green. So it's the left part of the loop and the left part of the loop. Then the plus is what kind of crossing the black and the red always make. And then you have A and B, which is how many twists you add here. Okay. And this, her theorem says that these are all the cases that you can actually, they are enough to actually get all possible knots and proteins. Okay, so it's cool. And I say, okay, why don't we do it for all trefoil proteins? And there are actually 12 cases for this double loop mechanism. And we did again the same thing. We generated a bunch of trajectories that mimic all of those 12 trajectories. So we have a bunch of those, and we generated also, the one loop. The consensus of research so far says that there are 4 mechanisms with one loop. So we generated also a bunch of random one loop trefoil knotted structures, and we took the pairwise distances, and as we expected, they were clustering together. Trajectories that had the same double loop mechanism and the same number of twists, they cluster together, and the one loop cluster together. And then we said, okay, let's take the representatives. This is what non-redundant says here. Let's take the representatives of all classes of proteins of sequence similarity and throw them into the mix. So we compare also pairwise distances using Knotoids and see what's happening. So we see, as expected, most of the trefoil proteins fall within the one loop, and this is consistent and coherent with what simulations and previous research has shown. And actually, most of the deeply knotted proteins fall here, if not all, which says that getting something more complex is, our results at least suggest that is out of the question, getting some more complex topology during the folding pathway of deeply knotted proteins. But what's more interesting, we have a full family of proteins, the carbonic anhydrases, that they follow somehow. There is an indication that they follow Flapan's suggestions, and that was okay. We say, okay, that's cool. So let's simulate that. So we did a simulation of that, but it was like a naive model. We didn't consider any interactions between amino acids, and we thought that this is quite enough to actually describe the folding mechanism. So what we did is that we unfolded the protein. Actually, our colleague Pavel Dabrowski did it. So he unfolded the protein in a nice way. I will spare the details, but I can come back if needed. And then he performed 200 simulations of folding of this protein, and it seems that it seems that either the red or green loop first, we have one loop forming first, then we have a second loop forming secondly, and then the tails are placed. So this more or less suggests that it doesn't actually follow 100% Flapan's suggestion, in the sense that we didn't observe any flipping, but we did observe 2 loops. So I think that our results suggest that at least carbonic anhydrases, they form a 2-loop structure while they're folding, and maybe that's because they're shallow proteins, and that's quickly the beginning of the simulations. But yeah, we may need more experiments on that. But in any case, Knotoids manages to capture this kind of geometric feature, and it's a quite sensitive measure that captures not only topology, but a bit more finer geometric structure that we can miss by applying pure topology. And that was something that made us quite happy because, yeah, we actually succeeded in leveraging the full distribution of the Knotoids, and we didn't just say, okay, we get the full distribution, then we just throw it away, just keep the predominant one. And this, as I said, appeared like yesterday on arXiv, and we will submit it in the following weeks. Hopefully, it will go well. And if I have some time, I will just say, just advertise our software. So our software here is Knotoid, and it was published in 2018. Time flies, in bioinformatics. And it's, if you're afraid of the command line, I'm sorry, but it's a command line tool, so you can input your Knotoid either in Gauss code or any kind of way, and it will compute the invariant that you asked it. And it can visualize, actually, the data. Everything that I showed has been visualized with Knotoid. Okay, yeah, some facts here is the first software that analyzes Knotoids and open curves. It has Knotoids on the plane, on the sphere. It's backwards compatible with literature in the sense that it supports analysis of closed curves also. And it has the infinity closure, as I said before. Suppose the PDB format for proteins, open curves in XYZ format. If you don't do proteins, you can apply it to your work. And if you're a mathematician and you just have a bunch of diagrams, you can plug in the Gauss code or the PD code format, which is a code format that Mathematica uses for the package knot theory. And we have the Jones polynomial supported, the loop bracket, and the loop arrow polynomial. And yeah, I mean, I will just skip those because we've seen them before. Everything was generated by Knotoid. We can actually do also coloring of structures and rotate them. This is interactive in the web browser. In the end, you get a WebGL object. And we can do some localization studies here that I didn't go through, but I will not. We can plot, available on all the popular platforms. You can download it from GitHub. It's open source. You can, if you feel like it, you can actually contribute. We will be happy. And the final thing I want to say about NodePro database, which is the 2.0 version that includes Knotoid as in the backend that analyzes Knotoids. To me, it's the most coherent database of noted proteins. It's updated every 2 weeks, and whatever new proteins are deposited in the PDB, they pull it and analyze it as knots and Knotoids and many other topological structures that they support. You can go process my structure, choose Knotoids if you have a 3D curve and you don't want to do it by yourself or in the command line, and they will send you an email with the results. And yeah, that's the details of the publication. And yeah, I would like. This is where I will stop. I think we are way over time, and we will not have time to talk about knots and chromosomes. But I think that was quite a lot of material that I presented, and I will have to thank, first of all, my colleagues in Switzerland, and Andre, Fabrizio, and Julien, that we worked very, very closely for the Knotoids pipeline, and the NotoEMD team, which is a bunch of very clever young people at Oxford, and Pavel Dabrowski, Tumansky in Warsaw. Yeah, Agnese Barbensi, Naya Yerolem, Oliver Vipond, and Barbara Mahler. All of them are, not all of them. Agnese and Barbara are postdocs in Oxford, and Naya and Oliver, they are PhD students, and I think they are graduating. Of course, the NotPro2 team, and you for your attention, and I will